finally, lab 5, we'll do a lab on Western blot analysis. Okay, hopefully you already reviewed the lecture of Western blot analysis. So again, what is Western blot analysis? Where is it used? It is a detection method where you can detect a specific target protein using an antibody that recognizes your target protein. Where is it used? When you try to identify your protein of interest from different protein samples. That's where you use it. And similar assay is called ELISA. ELISA stands for enzyme-linked immunosorbent assay. So same principle, Western blot, ELISA, same thing. ELISA is used a lot during the QC assay of downstream process. So basically, Western blot analysis, ELISA assays are very important when you try to detect your protein of interest during your purification, during your isolation process. Okay, so when you did the ion exchange chromatography separation of two proteins, right, equine myoglobin and bovine serum albumin, okay, two proteins were separated into two different peaks, okay, according to our chromatogram, and hopefully we will detect one of those two proteins using an antibody that recognizes that protein. In this case, we have an antibody, polyclonal antibody that detects a BSA protein. So we'll try to identify BSA protein and see if that is really BSA. We understood that those two proteins had differences in their sizes. We know how different their sizes are, but the Western blot analysis using BSA antibody will confirm that yes, indeed, that is a protein that you are looking for. Okay, so in this case, it's anti-BSA antibody. So against BSA, this antibody binds, but that antibody is made from rabbit. It's immunoglobulin G. That's what you're going to use for your assay. Okay, so questions to ask through this experiment. Okay, where do you expect to see your BSA among your ion exchange chromatography fractions? Okay, is that the first peak or second peak? What did your SDS page result indicate? Okay, can you tell the size of those two proteins? Okay, based on the sizes, can you tell which protein was located from which fractions? Can you also confirm your SDS page result through Western blot analysis? Okay, that's going to be the ultimate, I guess, identification if you use an antibody. So the process-wise, so last lab, not only you did the SDS page, at the end you also did the electroblotting, meaning you transfer the proteins from SDS page gel to a membrane, right? That's done blotting using electric current. Okay. After that, the first thing you need to do is blocking of non-specific binding sites that exist inside on the surface of the membrane. So how are you going to do this? You have a block of solution which contains saline, that's a buffer, and the detergent. Okay, it doesn't have any protein in it, but when you add solution B to it, solution B has a casein, milk protein in it. Okay, when you mix them together, that 20 ml volume will have the random protein that will seal up the random binding site on your protein. Okay, so you're going to incubate your membrane in 10 milliliter of blocking solution. Okay, that solution will have a lot of casein protein and it will bind up all the membrane binding sites. After that, you're going to rinse that blocking solution off of your membrane, okay, using water. Once that's done, then you're going to add primary antibody to your membrane. So primary antibodies again, rabbit IgG that binds with DPSA only. So that 10 ml solution. So you're going to incubate that. So you're going to add the antibody solution. I will give you 0.2 microgram per ml to 10 ml. So you're going to incubate your membrane with this solution for about 1 hour. So the concentration of the primary antibody stock solution is 2 milligram per ml, which is same as 2 microgram per microliter. Okay. Then how much of this do you need in 10 ml blocking solution? So in every 1 ml you need 0.2 gram. So then in 10 ml how much do you need? In 10 ml you need probably 10 times of that, which is 2 microgram. So how do you get 2 microgram? This concentration 2 milligram per milliliter equals 2 microgram per microliter. So what? So you need 1 microliter of that. That will give you 2 microgram of your antibody. So add 1 microliter of your antibody stock solution to the 10 ml of blocking solution. That will give you your primary antibody binding solution. Make sense? And then so incubate your membrane to the binding solution about how long? So 1 hour incubation, and after that wash the membrane, okay, by after getting rid of the binding solution, antibody binding solution, and wash it with wash solution, okay, about 5 minutes, 5-minute wash solution, 5 minutes, about 3 times, okay, to make sure all the primary antibody is gone. Once that's done, you're going to add a secondary antibody. In this case, the secondary antibody is made from gold. It's a gold antibody, but it recognizes rabid IgG, so it'll probably recognize and bind to the primary antibody where it's sitting right now. Not only that, this gold antibody is conjugated with the alkaline phosphatase enzyme at the end. Okay, so it's going to give you the signal you need. So incubate 10 ml of pre-made secondary antibody solution. The solution already has the secondary antibody added. Add it there, and then incubate about 30 minutes and get rid of the solution. And then you rinse the membrane just like step 4. So step 4, you rinse it 3 times, right? So yes, you use the same method to rinse your membrane, okay? And then finally, after that, you do a water rinse one more time, 20 ml of water, okay? And rinse it 2 minutes and repeat it twice, okay? So actually 3 times you rinse with water, okay? And then you add your substrate that will be broken down by alkaline phosphatase. There's a chromogenic detection solution, okay? So add 5 ml of the solution to your membrane, okay? And there's going to be a conversion happening, so it'll give you a blue color to the secondary antibody that has the enzyme alkaline phosphatase. So you will be able to see the band showing up as a blue color. So overall, it's going to be this process. So if you see the blue color band, you're going to stop the color development by adding water and rinse with water about 2 minutes. So target protein probably BSA, and then primary antibody anti-rabbit BSA IgG. The secondary antibody gold IgG recognizing rabbit IgG, and then secondary antibody has the alkaline phosphatase. Alkaline phosphatase substrate will be, converted into blue or purple product. So you can see that purple color of this protein band on your membrane. That's how it works. Okay, so that's it for the lab.